It now. So you might have received a prompt to accept recording for continue or to continue. And if you accept that, you should be able to continue. Did everyone get that prompt? Just to make sure that we are recording.
Yes.
Okay, well, that's good then. So once again, thank you all for joining. My name is Krishna. I work at the Gladstone Institute's Bioinformatics core. I will be leading the discussions today and tomorrow. We also have Min, Alex, and Ruben, who will be instructing different parts of the workshop. So on Wednesday, Ruben will be instructing a discussion on differential analysis of single cell RNA-seq data. And on Thursday, it will be Alex leading discussions on, or do you all see my screen? Not. So on Thursday, it will be Alex discussing network analysis approaches for single cell RNA-seq data. At any point in time, if you have any questions, please feel free to unmute yourself and ask. We have Min here. If you have any questions that I cannot answer or we need extra help with, then I will direct them to Min. And you can also save some of your questions if you feel that they are unrelated to our discussion today. For Thursday, we have 1 hour dedicated to Q&A. Although I will suggest that you please feel free to ask any question that you may have. I may direct some questions for to Thursday, but I think it is best that you do not assume that your questions are not relevant to our discussion today. Please feel free to unmute yourself at any point in time. I do not get offended, so please feel free to interrupt also and ask any question you may have. There are no specific questions. So our discussion today is on current practices in analysis of single cell RNA-seq data. Actually, what I would like to start with is the survey that I sent, and looks like not everyone has filled it out, but that's all right. We just want to get an idea of what the composition of our audience is, and it looks like there are some people who are not very familiar with our scripting. Maybe some of you have only taken introductory workshops, which is all right. We won't be doing hands-on coding in this workshop today, but I hope that even the person whose experience level is level 1 has at least attended the introductory R workshop that we conduct. And for those who have more experience, I hope there will be something useful for you to learn. It also looks like not many of us have experience with single cell RNA-seq, or some people have experience, are currently planning to perform single cell RNA-seq analysis, but people have no experience. Many of us do not have much experience with that. So that's all right. We have accounted for that. And also it looks like a lot of us are wet lab biologists. So again, our discussions, I hope, will be simple enough, or I hope, get into any statistical jargon to start making no sense. I hope it will be useful for everyone. And it looks like some people have also tested the script and it runs properly on their system. So I'm hoping that it will work for everyone. So just given our background, I think it is perhaps a good bulk RNA-seq protocol, or what is going on in a bulk RNA-seq assay is a good starting point for our discussion. Once again, before I proceed, I just want to point out that on all these slides, I have references at the bottom, and suggested readings. So in case you are looking to learn in your own time using our slide deck as a reference, you can do so with some guide from what we have in our notes at the bottom of the slides. So, okay, so what is going on in a bulk RNA-seq assay? Let's say you want to compare a diseased sample with, so a group of samples which you have collected from some diseased patients, let's say, and you want to compare it with healthy normal samples, and you want to identify which genes have maybe, let's say, have manifested a change in their expression levels. Maybe those genes are associated with the disease, and maybe targeting them will lead to some better therapies, etc. So maybe what you do in that kind to identify those genes, what you can do is you can collect a sample of cells from or tissue from your diseased patients and your healthy normal individuals participating in your study, or they could be cell cultures or they could be mice, and you collect the samples and you extract total RNAs from those samples, and you enrich for your target population of RNAs, which in many cases are commonly messenger RNAs. So you perform something like poly A enrichment, or to get polyadenylated mRNAs, or you deplete ribosomal RNAs and tRNAs, etc. And then you have a population of RNAs in solution which you are interested in studying. And since most of our dominant sequencing platforms, they sequence only DNA, so we have to go through an intermediate step of library preparation where we convert our RNA molecules via reverse transcription into complementary DNA molecules. And in the process, our goal, the library preparation also involves attaching or ligating adapter sequences to the ends of the cDNAs. These are meant these are added so that the library that you prepare can be sequenced on the instrument, because these adapters are needed for the cDNA library to work with the sequencing instrument. So once you have prepared that library, you'll send it to a sequencing center, such as maybe in collaboration with the Genomics Core or CAT Core from UCSF or somewhere or some other vendor. And those folks, they sequence your cDNA library, which gives out, basically files containing ACGT, ACGT, or whatever is the sequence of your cDNA molecules is. And there is one read per molecule. And after we get that file, our goal is to map it back to a reference transcriptome or genome to identify where those reads are originating from. So this is where this is the starting point of analysis, kind of. There are some other pre-processing steps which you have to do, but let's say we are doing this to map our reads back to a reference sequence. And then after we have mapped the reads, we tally how many counts are mapping to different genes. And that's is a starting point for later statistical analysis of the data. One problem one limitation of the bulk RNA-seq protocol is that we do not get information at the level of cell types. So for example, our original starting sample may be a mixture of multiple cell types. Let's say if you are studying brain tissue, there may be neurons and astrocytes and microglia and dendrocytes and other things which make up the brain tissue. And maybe the disease is impacting only one of the cell types and not all of them. And you don't really know before you start with your experiment which precise cell type is the disease associated with. So in that case, if you mix all the samples to all the cell types together, which is what you can do in bulk RNA-seq, then maybe the signal will be diluted, or maybe you will not have the precise pinpointed association of or identification of what is causing the disease, which is maybe our goal in our biological study. So for those purposes, it may be helpful to isolate the cells in our sample into single cells and then perform single cell RNA sequencing. And to facilitate this, there are many technologies which people have developed in the last 10 plus years. And one of the dominant players in the market is 10x Genomics. And on this slide, what we have is an illustration of the technology that 10x Genomics works with. So their instrument, or I think their instrument is called as Chromium. I'm not sure, that may be incorrect, but in any case, this is the way 10x Genomics instrument works is, first of all, what you will do is you will isolate your single cells using, let's say, enzymes such as collagenase, or there may be some specific protocols for the kind of tissue that you are looking at. and you apply those protocols to isolate the tissue into single cells, and then you load it at a low dilution, at some specified level of dilution onto a microfluidic device. And there are, along with the cells, there are these 10x barcoded gel beads which are also loaded onto the device, and the rate of flow of these beads and the cells and are optimized, or they have engineered it in such a way, they have engineered an instrument which controls these cell input and the bead input in such a way that finally these cells and the beads, they combine together in a way that there are drops created in a gel, sorry, in a water in oil emulsion, where there are drops of water, a nanoliter sized drops, each drop containing, hopefully and ideally, each drop contains one bead, one single bead and one cell. And these beads, they are not just balls, they are random common ordinary balls. These beads have something on them. They have oligos sticking out from them, and these oligos, they have uh, these are oligonucleotides which have a barcode, a 10x barcode, which is unique to each bead. So each bead will have a unique barcode sequence, and all the oligos sticking from the surface of this bead will have the same barcode, but different beads will have different barcodes. So, and then alongside the barcodes, there are also oligo DTs, which are going to prime the poly A tails of mRNAs. And when the reverse transcription happens, along with the mRNA sequence, the barcode of the 10x barcode on each bead will also get added to the cDNA sequence. So part of the cDNA sequence that will be generated after reverse transcription will come from the mRNAs in your cells, and a part will be the 10x barcode. And then there will be other primers and adapters, etc., which will be for the purpose of sequencing. So now using this technology, what we have is essentially we end up with the same cDNA library that we kind of get from a bulk RNA-seq protocol. But in addition, we also have the 10x barcode, which can help us identify which are the reads coming from a single cell. And we can group them together and make them represent a profile for a single cell. So that's the technology behind 10x genomics. Over the decade plus that we have of efforts in developing single cell technologies, people have come up with many innovative solutions. Some of them are not droplet based, as we have just discussed in the case of 10x genomics. Some of them use plates, microwells, arrays, etc. But as you can see from this figure, 10x or droplet based technologies dominate the field, probably. And one of the reasons for that is that they have a higher throughput. One can study a lot more cells with droplet based technologies, but other technologies also have their advantages and disadvantages. There is a good discussion by Dr. Eric Chow from UCSF. You can find it on YouTube, where he introduces many of these sequencing technologies for single cell genomics, and you can study that in your own time. Any questions on what we have discussed thus far? So one of the advantages of single cell RNA-seq, as I mentioned, is that we can study biology at the level of cell types. We do not have to mix them all together. We can, and also in some cases, like for example, maybe we are studying tumor specimens. And in those cases, it may be possible that there are some new cell types in the population which we don't really know of from before we started our study. So we can identify those new cell types. This is an application area where bulk RNA-seq may not be applicable. And then in certain cases, let's say we are studying the dynamics of developmental processes and development, especially of if it starts with a single cell, then well, you have no option. You have to study development at the level of single cells to study how the cells are differentiating and what changes are happening in cells as they are differentiating. So, and also, in combination with other newer technologies, newer additions to the whole suite of technologies for single cell genomics, such as spatial transcriptomics and multiomics, there is now a lot more, there are a lot more options now to study biology at a level of detail that was not possible earlier. Yet, when we plan our studies, we might want to also look for alternatives to single cell RNA-seq. It is maybe possible to achieve your project goals without going into single cell RNA-seq. For example, you may be able to sort your cells into subpopulations using, let's say, FACS or flow cytometry or something like that. And you can perhaps study your individual subpopulations now using bulk RNA-seq after you have sorted them. So it may be possible in some cases. So you may want to consider such approaches before just blindly jumping into single cell RNA-seq. Yet it may be possible. There are some limitations to single cell RNA-seq and some challenges. So one of the main ones is that it is more expensive than bulk RNA-seq, and typical studies are, because of cost limitations, typically underpowered. There are generally fewer replicates, and people also there are higher, more computational challenges when it comes to single cell RNA-seq data analysis. So you might want to consider that also when you design your study. In particular, many folks are not doing replication at the level of organisms. This is fast changing, but some folks have a misunderstanding that sometimes there's a misunderstanding that each cell is a replicate of another cell. That is true if you just want to make claims about one particular individual, what things are happening in that individual. But if we want to make claims at the organismal level, then we want replication also at the organismal level. And also, let's say we are working with cell cultures, which are, let's say, cardiac cells derived from iPSC cells, then we also want replication of our differentiation protocols to make sure that nothing fishy happened during the steps of differentiation. So for those kinds of consideration, you want replication of your single cell RNA-seq sample itself. Not one cell may not, the cells may not be replicates of each other at the organismal level. So there are a couple of questions on this point in the chat box, so I will just quickly address them. Can you discuss the underpower issues a little more? Well, I did discuss it a little bit more. If you have specific questions regarding, for example, Shiji has a question regarding how many cells are required for each experiment. Well, there is no clear, generally the recommendation from 10x Genomics is go for 6,000, 7,000 cells, but I think that is more about the cost or what people can afford. So if people have lots of money, then you can go for more. I think the current limitation is more about how much you can afford.
What
3-4 replicates, I would say. And
I was the one that asked about the underpower issue a little bit. I wonder if if I was to do this experiment and instead of pooling the animals, when I get the single cells into one pool, I kept the animals separate and then end up, for example, having 3 biological triplicates and each one underwent single cell RNA sequencing, would that address the underpower issue?
it will mitigate it, but it is hard to say will anything address it, because there are, in general, from what I have seen, many of the power analysis studies, they conclude that you need a lot more replicates than is practically possible, like 20, 30 replicates, biological replicates. Sometimes you will find that that is the conclusion from power analysis studies. And I don't think that is within the reach of most labs. So certainly 2, 3 is better than the what many researchers can afford. So if you have budget for 2, 3, I think that is a good progress. I don't think that anyone can say that power issues will ever be completely avoided. You would have all the individuals in the world.
Thanks.
Yeah. And you are also welcome to ask this question to Ruben, who is more knowledgeable in power analysis and all that, or on Thursday in the Q&A session. Thanks for the questions. And there are, besides these limitations, also there are other technological limitations. So for example, one limitation that some studies have pointed out is that currently single cell RNA-seq should not be utilized for alternative splicing studies. And that is because of the many technological limitations of the single cell RNA-seq, which lead to a lot of genes just not being observed. And sometimes people have come to maybe misguided or misled into some observations that are perhaps not true. So for example, here in the references, you can find a few which were, in particular, this one from some folks over in UK, University of Cambridge, and then this one from a group in Berkeley. They discuss how there may be some misleading or wrong conclusions, one may get to if they are using single cell RNA-seq for alternative splicing studies. Yet there are also, in the last 5-6 months, there have been some bioarchive papers to discuss how one can use single cell RNA-seq for alternative splicing studies. So you are welcome to look at all that, but I think the word is still not final. Um, just be cautious if you are applying single cell RNA-seq to alternative splicing studies. But also because of low sample size and many of these many of the technological limitations, many folks recommend, and it is also something that I agree with personally, that we should use single cell RNA-seq as a way to generate new hypotheses, but they require further validation using other independent approaches, maybe low throughput approaches, to make sure that what you are finding is actually real biology and not just the technical biases of the instrument and the technology itself that you are calling biological discovery. So be mindful of that. And the reasons for single cell RNA-seq analysis being more challenging, some of them are listed here. One is that data is sparser than bulk RNA-seq, since we are dividing almost the same level of sequencing depth over many more single cells. Each single cell, what the observation has been, that there are lots of genes which are just not observed. There is not a single read which maps to many of the genes, and that is because we have not, we cannot sequence deep enough. And also, if we want, let's say, 20 million reads per cell, then we will, and we have 6,000 cells, then you can see that there will be a lot of money that you will have to spend on sequencing. The data is high dimensional. We observe thousands, let's say there are 30,000 genes. So we will observe all of the genes in probably we will observe all of the genes in one or the other cell. Maybe we will not observe thousands of them, but majority of them we will observe in one or the other cell. So and we end up with the counts matrix, which is of course almost the same size as bulk RNA-seq assay, where there are 20 plus thousand genes that we are working with, but there are also now many more thousands of cells. And there are few replicates, if any. And then there is, of course, technical noise, which is more severe in the case of single cell RNA-seq. There are batch effects, and then oftentimes also what we are looking at, let's say we are looking at the expression profile in developing or differentiating cell populations, then in those cases we can expect there to be some nonlinear expression profiles across time points. So to study all those kinds of things, then there are more challenges when it comes to analysis. So we want to discuss all that. Our introduction went on a little bit longer than our planned discussion, but that's all right. Our goal for today is to get to cover these first 5 points. I think we can do it adequately in our remaining time of 1.5 hours. And then tomorrow we want to cover the rest of the material. This is an approximate time distribution. It's not, compulsory that we stick to it. So our goal in this workshop today is to give you an overview of current practices in single cell RNA-seq analysis, and highlight the assumptions and limitations underlying some of the methods that we will be using, and understand their relevance and impact on downstream steps. So I have shared this R Markdown document with you all. I hope everyone can see my screen adequately. This is implementing the standard workflow using a tool named as URAT, which I will introduce in a few slides, from the Satija lab over at New York. And if you have attended our recent workshop on introduction to R, you will note, as I mentioned there, that many of these functions, they have logical English word type names. So for example, we will start with creating a URAT object, then we will identify the mitochondrial proportion, the proportion of reads mapping to mitochondrial DNA in our cells. And then we will make some plots, Violin plot, scatter plot, and we will normalize the data, find variable features. I will introduce what all those things are doing. But these functions, they have very English word type names, and they are well described in this tutorial on this website of, from the Satija lab. So you can study it in your own time. Running them is not a big deal. I think what is a major challenge and many people struggle with is what is going on with these functions and what they are actually doing, because we just don't want to treat all these packages as crystal ball, where we are taking our data to, let's say, a fortune teller, and they are doing whatever they have in their system, and then we don't really know what is going on. So our goal in this workshop is to develop some understanding of what is going on under the hood, and that is what our discussion will be focused on. If you cannot run these commands, that is all right. If you just want to listen into the discussions, that's all right too. And I will introduce all these commands as we move along with our discussion. So in general, this is again, a typical workflow, and a real analysis in the real world may deviate from it in many ways. But what we have discussed is one starts with their experiment design, prepares a library, performs sequencing, and then once one gets the data from sequencing, then the first task is to demultiplex in case you have multiplexed multiple samples together. Then you demultiplex them, and then you do the alignment to a reference sequence, a reference genome sequence, and then you count the number of reads mapping to different genes, and then you get the count matrix. And then you have to do some quality control to filter out poor quality cells, normalize the data, perform some dimensionality reduction and clustering of genes, and perform differential analysis, and then lots of other things that can happen downstream. So I will introduce all of these things, and we will start with pre-processing of raw sequencing data to count matrix. This part we have not provided any hands-on guide to, but you can, perhaps hopefully you can use our slides as adequate guide in case you need to implement pre-processing on your single cell RNA-seq data. So let's say you prepare your single cell RNA-seq library, and for which you can consult with the Genomics Core or other folks how to prepare that library. And then you perform deep sequencing, example in collaboration with UCSF CAT Core. Now the data will be generally large, so of course that is the reason why we cannot do it in this duration of our workshop. But they will send you, after they have done the sequencing, let's say UCSF CAT Core, they will send you an email with instructions on or where you can find your data on a remote server, and then you can log into it and download from there. We have tutorial on Winton. Again, you can find the link on how to move data from CatCore to your your own directory on Winton, and you can get your files there, and then you can pre-process it using some tools such as Cell Ranger. There are multiple tools out there, but Cell Ranger is one which is from the 10x Genomics itself. So that is pre-installed on Winton, along with other tools such as Callisto, I think is also installed on Winton already. But these different tools have their own pros and cons, and maybe you can work with CellRanger to start with, but you can also look into these other tools. Callisto in particular, it is much faster than the others, and UMIs I think is much more flexible. And in terms of what you can optimize the parameters, etc., to a higher level of flexibility than the others. I think that is the selling point for ZUMIs. So there are these different tools for pre-processing your data. After you pre-process, we will be discussing in the context of cell ranger based pre-processing, and you can look into other tools in your own time. And after you have pre-processed your data, then there will be some QC files which will be saved by cell ranger, and we will inspect them and try to interpret them today. So cell ranger pipeline, it is available on Minton from the CBI software repository. There are three steps which typically you will need to implement. One is CellRanger make FastQ, which is to convert the BCL format files to FastQ files. The FastQ files are the ones which have the sequences. You can open it and you can see that there are a CGT and the quality scores for each of the base calls. And we discussed this in our Intro to RNA-seq workshop recently. So I'm skipping all that. But if you are interested, you can look at our Intro to RNA-seq slide deck. This will not be that important for our later discussions. So you run this demultiplexing tool, and then you align using cell ranger count. And then finally, if you have multiple samples, then you aggregate them using cell ranger aggregate. And the output from the cell ranger software is a BAM file for each of the sample. BAM files are where the alignment coordinates of the reads are stored. So for example, if you have a read, where does it align to in the genome? So the start and end coordinates of the read with respect to the reference sequence, which is the chromosomal sequences, that will be stored in the BAM files. There is a web summary HTML file, which we will look at in the next slide. And then there are count matrices, which are storing the data for, let's say, 20,000 genes. And if you have done 6,000 cells, if you have prepared your sample for 6,000 cells, then that will be the number of columns in your count matrix. For each gene in each cell, we will have a number. And there are two kinds of matrices which Cell Ranger will save: unfiltered matrices, which will contain all known good barcodes. So the barcodes here are referring to the 10x barcode that we discussed in our previous slides. And this will include both the barcodes which correspond to cells and empty drops. And we will discuss what that means in the next slides. There will also be filtered matrix, which will store only barcodes that appear to be associated with cells and not empty drops. And then these data will be organized in three files, and then there will be some results from secondary analysis, such as dimensionality reduction and clustering. Generally, people ignore them in the favor of downstream analysis using SURAT or ScanPy or other tools. And CellRanger also saves a loop file, which you can load with a loop browser to look into the data or to look at the QC reports and also secondary analysis in some detail. There is also a molecule info file, which is a compressed format, saving information about each molecule and where it goes to, maps to, etc. So you can again explore that in your own time. All of these files I will not discuss, but I will discuss some of them, which are generally the files that people are looking at. So for example, this web summary.html, it is a useful output from Cell Ranger. It gives you information about some useful sequencing metrics, such as number of reads that were there, that the Cell Ranger could disentangle and assign to each of the samples. How many of those reads have valid barcodes because of sequencing errors or some other reasons? Maybe some reads will not have the barcode sequence that Cell Ranger tool can identify. It has a catalog of all the barcodes that the beads have. So maybe some of the barcodes, some of these reads will not have any valid barcode. In general, you will want most of your reads to have valid barcode. And then there are also unique molecular identifiers on each read, which is there to disentangle or deduplicate PCR duplicates. So when we are preparing our sample, since we are starting with a very low samples, with a very low, sample size, or our each cell will have only so much RNA, and we have to amplify that signal, which is done using PCR. But in that process, what we can end up doing is we can end up, amplifying different molecules to different levels because of the biases of the PCR technology. And what we ideally want is to count how many copies of individual molecules we have observed in our cells. So that is done using a unique molecule identifier, which is also a part of the reads. So reads that have the same UMI are PCR duplicates of the same molecule. So that's what UMI means. And we want ideally to have lots of our reads to have valid UMIs. And then there are other things that it informs us about, like how many reads mapped, how many reads mapped to exons. So again, if we don't expect pre-mRNAs in our population, then perhaps, or we don't expect much of introns, then maybe our read should map mostly to exons. So those kinds of informations you can look at in this web summary.html file. There is also information about how many cells you were able to capture, how many reads were there in cell, and all those kinds of things. And there is this an interesting looking plot on the top right, which is called as barcode rank plot. So let's quickly discuss that. So as I mentioned, when we are preparing our sample using the single cell or 10x genomics droplet based method, then our goal, ideally what should happen is that each of these drops that we create in this emulsion, there will be one bead and one cell per drop. But there may be some drops which will have no cell. But it does not mean that they will not have any RNA, because there may be some ambient RNA, because from other cells maybe, or from your sample preparation, maybe cells are leaking some mRNAs while you are preparing these drops. So in that case, you may end up with a situation where there are some empty drops, and typically this is the case. There are always some empty drops, perhaps lots of them. So you want to identify and remove those empty drops, because they will also contribute some reads. When you perform your library preparation, after you have prepared these drops, you add the same cocktail of enzymes and nucleotides, etc., to all the drops, and they have the reverse transcription and library preparation happens in all of them. So some of the drops will give you reads, but they will not be cells. So how can we identify that? And that is what is the way to do it is based on how many unique UMI counts you get. UMI counts is the unique molecular identifier count. So after you have deduplicated the reads, you look at how many total UMI counts you have per drop. So each drop is represented by a barcode, and each molecule that you observe is represented by a UMI. So how many UMI counts you have for each cell or for each barcode, that is what is plotted in this barcode rank plot. First, they sort the barcodes by the number of total UMIs they have, and then they plot it. The basic idea is that the drops or the barcodes which have very few UMIs associated with them, they are perhaps empty drops, because cells will have a lot more RNAs than the empty drops. That is the assumption here. And by looking at the barcode rank plot, the goal here is to identify this sharp drop in the number of UMIs. So as you start, so if the highest UMI count in any barcode is, let's say, 100,000, And then there is then it keeps decreasing from there, because that was, of course, on the leftmost point we have the highest value, and then you see a decrease. But maybe there is a continuous decrease from one cell to another. You expect there to be a gradual decrease, but after you shift from cells to empty drops, then maybe there will be a sharp decline in the number of UMIs associated with a barcode. So the goal is to identify this knee kind of thing and cut off, use that as a cutoff. So everything to which has more UMIs, all the barcodes which have more UMIs than this cutoff will be called a cell, and everything with fewer UMIs will be called an empty drop. I hope this makes sense. So there is a question from Amin Ali. What would be a good threshold for mean reads per cell? Well, ideally, what we are looking for is mean. We are looking for, what would be a good threshold for mean UMI count. So there may be a lot more reads than UMIs in a cell, because reads are before deduplication. We are, at least for our discussion, we are calling them reads before deduplication. So ideally, we want a lot of UMIs. And how many is a good level of UMIs? Think, you would want thousands of UMIs, millions maybe per cell, but that is not possible in single cell RNA-seq. So you work with whatever you have. Again, there are more. The limitation is more of the cost, and with given the cost 2,000, 3,000 UMIs per cell, at least the current status quo is that is considered, well, I think, good. But that is because of the cost constraints, not because that is good, really good. Ideally you would want perhaps millions. We will discuss again. I'm addressing some of the questions that we have in the chat box. Thank you all for asking those questions, and again, feel free to unmute yourself at any time. Or we will get to doublet rates in our later slides. Is further towards the right. Yes, we will get to that also. So there is a question from SUP regarding the UMI and barcode, which I think I can discuss now, and the other questions I will address in the future slides. So let's go back actually a few slides and look at this, what we are attaching to our cDNAs. So there is the 10x barcode, but there is also a unique molecular identifier. What this is a unique sequence. So every time a unique RNA molecule is reverse transcribed and its signal is captured into a cDNA, we will also attach to it a unique molecule identifier, which is basically again a DNA sequence, ACGT or something. It is different for each of the oligos sticking from the surface of this bead. And what it will help us is, let's say now after we have prepared we have done the reverse transcription to get our cDNAs, then we will amplify those cDNAs using PCR technology. And what PCR will do is it will create many more copies of each of these cDNAs. But all of those copies will have the same UMI if they come from one single molecule. So what is done after you do the sequencing, you identify, you collapse all these copies of DNA of the reads that map to the same site and have the same UMI sequence. They basically are coming from the same molecule, which was amplified many times by PCR. So we want to reduce them to one single count, because that is basically, we want to count the molecules, not the reads after amplification. So that is the goal being served by UMI, and barcode is for the cell. So I hope that answers the question from SUP. Can you please confirm? Okay, thank you. So once again, all questions are welcome. We will meet the we will hopefully not exceed the time, but anyway, I think our discussion is important. So all questions are important here. And if you are asking these questions, you are also asking them for other folks. So please, I do welcome those questions. Okay. So in this analysis of identifying empty drops, the assumption was that UMI counts for cell-associated barcodes is much greater than UMI counts for background-associated barcodes, because the background-associated barcodes only have ambient RNA, and that ambient RNA is actually also present in the cell-associated barcodes. So the cell-associated barcodes should have a lot more UMI counts because they have the real population of RNAs, the real juice there, while the others are empty. But there is, again, this assumption um, may not be great, because there may be some cells which may have, like, low mRNA content. All the cells are not the same size, right? So some cells may have fewer mRNAs, or some other reason they have fewer UMI counts. So how can we increase our or improve our identification of barcodes associated with cells? So empty drops is a method which is now also implemented in Cell Ranger, I think version 3 onwards. What this does is, after there is a cutoff based on the total UMI counts, it looks at uh, the barcodes which have the lowest UMI count. So it will look at the bio reports, at this tail end with the fewest UMI counts, and it will create an ambient RNA profile. Like, it will use those to create the levels of different RNAs present in the empty drops, and that will represent the background profile of the RNA species in empty drops. And then the drops, which are at the extreme, which are very close to what we have called as empty drops, but they are very close to the cutoff of where we are not sure. It will compare those drops with this background profile, and if the profile does not match or seems very different, then maybe those drops are also containing cells. So that way there is an improvement in the identification of cells versus empty drops, and that is a part of the 10x pipeline now. But also you can do this in R or something. So Cell Ranger utilizes some of the tools that we have also discussed earlier in our Intro to RNA-seq workshop, such as Illumina's BCL2 FastQ and STAR. And the pipeline for Cell Ranger is kind of similar, where you start with the FastQ files, you trim adapters, map reads, tally counts, which we have also done in our Intro to RNA-seq workshop. So anyway, now there are some considerations which I'll just point out. As I mentioned, there are two kinds of files which Cell Ranger outputs: filtered files and unfiltered files. The filtered files do not have what are called as empty drops by Cell Ranger, while the unfiltered files have everything. So which one to use for later analysis? Now, as I mentioned, Cell Ranger, the latest version, has this new method based on the comparison with the ambient RNA profile to increase the efficiency or the classification performance for empty drops versus cells. But you may want to do it in R in case there are newer versions of this tool, empty drops, then, or other tools. People are this field is a very active research field, and there are lots of methods being published every month or every day. And also, I believe, for identifying empty drops versus cells, I think there is still some work going on, as well as while the technology improves or changes, the methods for classification of empty drops versus cells can be different or improved. So you can look into that, and perhaps you may want to use the unfiltered output from Cell Ranger and run empty drops. By default, at least what we have been doing in the core is running Cell Ranger 3 or later versions, the latest versions of Cell Ranger, and not running empty drops in R, but you may want to look into that. And another reason for there to be fewer cells, besides there being heterogeneity in the cell sizes, as is depicted in this or discussed in this paper from the, I think, Arjun Raj's lab. at UPenn. Um, there are other reasons also possible, and in particular, the example given on the Cell Ranger website is of heterogeneous population itself, where you are having multiple cell types in your population, and some cells are bigger than the others. For example, tumor cells in a population may be much bigger than the lymphocyte cells, which may be infiltrating your tumor specimen or tissue. And then, if you are interested in those lymphocytes, you don't want to just remove them because they have very low UMI counts, because they may be real cells. So for those kinds of cases, again, empty drops type algorithms are useful. Another consideration when you will run this software on your own, you will find that there is an option to set downsampling of reads when you are aggregating batches together. So if you have multiple samples or multiple batches that you have prepared, as for example, someone was just mentioning that they have biological triplicates, in that case, it is possible that different samples will have different depths of sequencing, which may be for various reasons. But it is possible that there will be different number of total counts, read counts in different samples. So Cell Ranger provides an option to downsample the reads to make the total number of mapped reads equal across the samples. And this is again to mitigate any further complications and analysis that may arise because of different depths of sequencing for different samples. So you want to equalize the level of read counts before deduplication in different samples, because the read counts before deduplication are representative of the sequencing depth, not the UMI counts. Nevertheless, there are approaches which also allow you to downsample and equalize the UMI counts across samples. That is not the preferred or recommended approach to equalize UMI counts, but for some analysis, if you are applying some tools, it may be required, so you have to read it on your own. And in particular, uh, the latest development from the Satija lab regarding SURAT, or how to analyze data in this pipe tool that we will be discussing, based on their development in this paper in Genome Biology in 2019. They recommend that one should not be equalizing or downsampling UMI counts. So you may want to look into that. Or even, read counts. You can have different levels of sequencing depths in different samples. And the technology or the method by Hifmeister and Satija, it is supposed to account for that and correct for that level of differences. And then another consideration which people often have is, where do I do my analysis after I have run Cell Ranger, R, Python, or Galaxy? In general, at least my understanding is that different tools, they have different strengths, and there are different labs which prefer. Some labs prefer R, some labs prefer Python, and there are some people who prefer Galaxy, and there are good solutions in all of these. And I have again provided some links in the bottom of the slides, which you can study in your own time to identify good approaches in different languages. In general, you may need to run your analysis in different languages, both R and Python maybe. Surat and Bioconductor Monocle. These are popular tools for single cell analysis in R. Bioconductor is a collection of packages. Surat and Monocle. Surat is from Satija lab. Monocle is from Trapnell lab. And then there are also implementations in Python, such as ScanPy, which are getting more and more popular, especially because the format of data storage, the Loom format supported by ScanPy, is becoming more popular for data sharing between different labs and communities online, etc. So because of that, ScanPy is also becoming more and more popular, or perhaps it is already equally popular as Surat and Bioconductor and others. So there are different, these packages, all of them have different, some of them will have their unique algorithms simply available in them. So, that they may make them better for, or, more useful for your purpose. So you may want to choose that. We will be using Surat for our discussion today because it is not possible to discuss all of them. But what we will be discussing is kind of a standard workflow that all of these different tools are implementing, and we will try to keep our discussion broader than one particular tool so that you can work with an understanding of what is going on in each of those tools. Because once you have that, running any of these tools is doing a few installations and writing very English word function names and running analysis, as we discussed in our intro to our workshop. So I will address a question from SUP. It looks like there is more than one knee in that graph, and what does the second knee represent? Thank you very much for the question. I actually did mean to discuss it in a little bit more detail. So sometimes there may be multiple knees, in which may be representative of, where is this? Which may be representative of, let's say there are two different cell sizes. Let's say there is a heterogeneous population that you are looking at, and maybe some of the cell types are generally giving you a higher UMI count, and the others are giving you a second lower level of UMI count, but still more than the empty drops. So that may be what is going on with the second knee. In some cases, when you have your sample fails, you will not see a clear knee like this. You will have more rounded curve, and that can be because of a compromised sample, or there may be very few barcodes associated with cells, let's say hundreds. That is not ideal. So in those cases, sample may be failing, and as this is showing, heterogeneous sample may have multiple knees. So again, you have this link to discussing all that in the slides. Thank you for the question. Okay, so let's proceed. So I have discussed now some of the considerations for pre-processing of the data. And now after we have done this pre-processing, we will have a counts matrix, from which point onwards, we will either be using R, Python, or Galaxy, as we just discussed, to analyze our data. We will be using R today because we have just had an intro to our workshop. And the first step will be to load the data in R using scRNA-seq. Again, you may want to analyze it using bioconductor or monocle or something else. We will be using scRNA-seq today. And I will just quickly introduce the data that we will be loading in scRNA-seq and what scRNA-seq is. So the count data that we receive from Cell Ranger output, it is organized in 3 files. There is also one compressed format which is storing all these together, but we will be loading it from these count matrices, which are stored in 3 files. So the reason to store it in 3 files is given here. So first of all, the files I have shared with you, they are the kinds of files we will be using. So for example, after you have downloaded the data, and if you have uncompress the zip file that I shared with you, you will find that there is a data folder, or perhaps it is called as PBMC3K_data. And you have that data folder. In that, you will find these 3 files, matrix .gz file. So these gz is representing another compression format, which is popular in bioinformatics. And mtx is representing it is the extension for matrix market exchange format. And here on the right, I've represented what this is about. Basically, this format is favored for sparse matrices, which have mostly 0 entries. So for example, let's consider our count matrix in our study. Let's say we have 20,000 genes, which we have observed for 6,000 cells. So then there is an order of 10 to the power 4 genes that we have in our data, for which we have data. And there is an order of 10 to the power 4 cells that we are also working with. So in total, there will be 10 to the power 8 entries, for each gene, and each cell we have 1 entry. So there will be 10 to the power 8 entries. But in typical single cell RNA-seq in current times, we have only 10 to the power 6 non-zero entries approximately. So it means that there are 100 times more entries. If we are storing zeros, then there will be a lot of zeros that we will be storing, 0 entries. So maybe we can do better or to save some space, and that is the purpose that is being served by the matrix market format. The way it works is there are 3 columns in this MTX file. If you want decompress it and you open it in a text editor, you will find that there are 3 columns. The first column is representing the row number of the gene. So each gene will be assigned a row number, and each cell will be assigned a column number. And the first 2 columns are representing the row number, the column number, and the third column is representing the value in that column. So, and the first line of this table is representing the total number of genes, the total number of cells, and the total number of non-zero entries. And the next lines are storing the values in a row, column, and the entry. I hope this makes sense. The information about which row number corresponds to which gene is stored in another file, and also similarly for the column names. So there is a file called as features.tsv.gz, which is storing the name for the genes in each row. So each row is corresponding to one gene, and there are gene symbols and the ensemble IDs or other IDs for the genes, and it could be other features also. And the column names, the barcodes, each barcode is representing one cell, and the names of those or the barcodes is saved in another file, which is called as barcodes.tsv.gz from the cell ranger output, but it may be called something else if you are downloading it from a public repository such as single cell expression atlas. I think there they have extensions such as column names or something like that. So you will also notice that the barcode is each barcode is a DNA sequence, an oligonucleotide sequence, and it has a suffix at the end, -1, -2, or something like that. The suffix is representing the sample ID. So when you run the cell ranger to aggregate your multiple samples together in one counts matrix, then each DNA barcode may be observed in multiple samples, but we want to keep each sample separate. So there is a suffix at the end attached to the barcode, which is representing each sample or GEMUL. Any questions about the data that we have shared? You all should have these 3 files if you are planning on running the hands-on analysis. Once again, folks, we are doing good on time. There is no rush. So the data for this workshop is on mice, from a mice study, or something related to tumor. We are not interested in the biology of this data. There are some features with this data. There are 2 groups and multiple time points, and there are replicates, and the analysis led to some new cell type identification. We are not interested in the biology of this data. I have not studied the paper in detail either. And unfortunately, I did not realize it initially when preparing this data, but we selected this data because it has some of the characteristics which will be useful for discussions in our third session, which will be led by Ruben. But this data is not from a droplet-based technology. It is not a 10x data. It is a SmartSeq data, which is a plate-based technology for single cell profiling. Apologies for that. If in general, when you are doing your analysis, your methods for analysis and the cutoffs that you choose, etc., may depend, or ideally would depend on the technology involved, and different technologies have different biases. So maybe you will want to study it in some detail on your own. What the recommended best practices are for that particular technology. For our purpose today, we are just looking at this data as a counts matrix, and we are not interested in the specific sources of what those the technology, etc., or the biology, etc. So apologies for that difference in our discussion and the data that we have chosen. In future workshop will replace it with droplet-based data. Anyway, now the next step that we will currently now run in R is to load the data using the read 10x function from the surat package and create a surat object. And just for your information, because sometimes folks get thrown off by the name surat, it's not a very intuitive name. So just for your information, this name surat is after the name of a 19th century French artist, Georges Seurat. They invented or popularized a technique of painting where they would paint contrasting, dots in contrasting colors, and from the contrast of the colors of the dots will appear the image that the artist wanted to convey in a style called as pointillism or something. As you can see on the right. In our case, these dots, which we will plot, will be different, which will be single cells, and we will make some scatter plots such as the UMAP plots, which you may have seen in some of the literature. And from those, the biology which will emerge. And with that kind of poetry in mind, the authors of Surat named their tool Surat. And our goal today is to run this tool, and we will use it for analysis. So I have shared with you this R Markdown document, and I will run for each step after we have discussed it, or while we are discussing it, I will also run the corresponding step in R. This is an R Markdown document where each of these grayed areas are representing code chunks, which we will be running. So you can run them by hitting this play button or the run button. And the first step is we will load these libraries: dplyr, surat, and patchwork. Dplyr is for some convenience of summarizing some of the data, some objects, and finding their summaries, which we will use later on. Patchwork is to put multiple plots together. Again, we will use it later on. And surat is the key software package which we are using for analyzing the data. So we will load these packages, and then we will load the 10x data. So it is stored in a directory called as data. It should be in the same location as the R Markdown document. So for example, my RMD file is in the same place as my data directory. So you should all see that there. And so if you have the data in the same folder, you can also set your working directory to current work source file location. If you don't do that, then it may also be throw an error, because R needs to know which directory you are working from. Again, I'm assuming that you are familiar with R, and we did discuss some of these things in our introductory R workshop. So you want to set your working directory to source file location. So when you do that, the R will set the working directory to the directory where I have the HTML file and the data directory. So what we are doing now is reading the 10x data from this directory named data using the function named as read 10x, again a very English word, and we are passing it to the counts field of this function create surat object. I will load the documentation for this function. So you can take a look at it in your own time, but the counts is where you pass the 10x data read in the matrix format, and then you project is a name which you give for your project. It could be something, it should be something meaningful. Here I just named it hello SC world, but it can be whatever name you want to give it. Minimum dot cells, it will, while loading the data, it will also implement a filtering of the data. All of the cells in our data is perhaps not useful for later analysis. Some of the cells may have very few genes. Maybe you did not observe counts for many of the genes, like maybe some and some genes will not be observed in many of the cells. So again, those cells which have very few genes observed and those genes which are observed in very few cells are not useful. So we will delete the rows and the columns associated with those genes and cells. Features is a jargon used in data science for something that we are observing. In this case, we are observing counts for genes. So they are our features. In other cases, they may be peaks. They may be, if you are doing single cell attack-seq analysis, they may be again, regions of genome. So features are basically genes in our case. So we are specifying, when we are loading this data, we are specifying that we don't want to keep genes which are observed in fewer than 3 cells, because they are not useful, or they are not going to be useful for our analysis, or we cannot say anything meaningful about those genes from our analysis at least. And then there are some cells which will have, let's say, very few genes. A cutoff, a popular cutoff in SURAT is 200 to start with. Any cell which has fewer than 200 genes will be removed. But you may want to look at some diagnostic plots, which we will generate in the next steps, and determine these cutoffs based on your observation of the distribution of number of genes per cell and UMI counts, etc. So you want, when you are creating this SURAT object, you want these cutoffs to be something very relaxed and liberal, not too stringent, because after you have created the SURAT object, then you can look at the diagnostic plots and then set better, more stringent cutoffs. While loading, we want some cutoff which is very liberal, but still, you don't really want genes which are observed in less than 3 cells. So that's something easy to remove. And here I have this extra parameter, which is names.dlim is equal to null. And that is because if you look at our data, then in our data, I will just unzip it and open it in text editor. So you can see that I downloaded it from online repository, and it does not have the barcode names as we would expect. And also this is smart seq based data. So I think there may be some difference because of that. But these are not things which we want to read. In general, the names field, the names of the barcodes, if from certain analysis, they may be in this format: barcode name, followed by cluster identity and cell type. So if things are separated by an underscore, SURAT assumes that the names of the cells are in this format: barcode underscore cluster underscore cell type. While in our data, this is not the case. So I am specifying that by specifying names.delimiter, which is by default underscore, I'm specifying it as null, which means that do not try to parse the read names or the barcodes to identify things like barcode, cell type, etc. I hope this makes sense. And we will hit this run button, and it will take a few seconds, and the data will be loaded. In the meantime, let's just see what we want to discuss. So the SURAT object that will be created here, let's give it a few, it is there. Now, after you have that SURAT data, you can look at it. If you just press data, then it will tell you that it is an object of class SURAT. It is a SURAT object, which has 21,000 plus genes observed in 6,000 plus cells. And there is only one assay. You could have RNA-seq and attack-seq and all other things which you can combine together in one SURAT object. We will not be discussing things like that. We can look at the structure of this data object using the function str in R, and it shows us that there are many slots. These are called as slots, assays, and then within that there are slots called as counts, data. There are these different slots which are storing different aspects of the data in one place. And also, as we perform analysis, everything will be stored in the same SURAT object. It will be like one, in physical terms, if you were collecting all your files associated with something in one folder or one bag, then that is the kind of thing going on here. We have one data object where we are storing all the things that we have relevant to that data. And our counts are here in one of the slots. So there is metadata which we can look at. For example, metadata field is storing the number of the rows of the metadata slot are the cell IDs, which are the barcodes associated with them, or the cell IDs in this case are not barcodes, but they are the cell IDs, and the number of RNAs that are observed in that, and the feature total number of genes for which they have we have non-zero counts. Here we have these fractional values, which is again perhaps because of the poor choice of data. It looks like the data is already normalized or something. So please forgive that for now. We are again not interested in the biology of the data. So we just want to run the tools, get an experience of running the tools in our hands, and discuss them using our slides. Any questions regarding what we have discussed thus far? Just quickly, that SURAT object can be converted to other formats required by bioconductor and ScanPy, as I previously mentioned. You may want to run your analysis using multiple different packages, and we are discussing them broadly. But if you do need to run your analysis in other tools also, then and have a mix-up of tools, then you may need to export your SURAT object into formats supported by other tools. So SURAT object is for SURAT. ScanPy requires a Loom data format. Bioconductor works with, I think, single cell experiment. So you have options available for that. You can look at this Vignet for conversion between data formats from from one tool to another using SURAT functions. Any questions regarding whatever we have discussed thus far? So what we have done thus far, hopefully the running the tool also worked in the R environment for all of you. Hopefully my explanation of the code was also clear enough. We have now looked at demultiplexing of the data, loaded the counts matrix in R, and now next step is to perform some more quality control of the data, check the, and filter out more poor quality cells, if any. So let's get on with that. So previously, someone also asked us a question about doublets, but that's where we are moving. So first of all, not all barcodes represent good quality cells. Not all genes are detected in all cells. So, and there are also perhaps some dead or decaying cells. So we want to filter out, cells which are maybe dead or decaying, or there are also going to be instances where instead of there being one single cell in each drop, we end up with multiple cells in each drop, which are called as doublets or multiplets. So we want to identify those things and we want to filter them out. And the way to do this, one way is to look at the unique molecules in a cell. So for example, if there are a lot of UMIs observed in a cell, maybe that is because those barcodes, associated with what is looking like one cell, maybe actually multiple cells. If multiple cells end up in one drop, then they will all receive barcodes from the same gel, from the same bead, or from 10x, and they will all look like one single cell. So maybe that is, maybe we won't want to carry them forward in our analysis because doublets, though they are not single cells, so we want to filter them out. Maybe some of the cells will have a high proportion of genes mapping to mitochondrial genes, which may be because those cells are dying. It has been found that or decaying, cells which are decaying, the cytosolic RNA diffuses out of the cells faster than the organelle associated RNAs, such as for mitochondrial genes. So RNAs from mitochondrial genes, if they are associated with the mitochondria, they may be preserved within the cell membrane more than the cytosolic RNA, so leading to a higher proportion of mitochondrial genes. Or there may be some other things happening. But that is my understanding of why there are higher proportion of mitochondrial genes. So by looking at these metrics, we can hopefully identify poor quality cells and filter them out. So that is what goes on in the next step of the analysis, filtering of poor quality cells. So first I will describe this code chunk, or maybe what I can do is I can run it by hitting the run button, and I can describe it now in parallel. So what we are doing, we created this data object called as data. And to that, what we are adding in the metadata field, we are adding one column called as percent mitochondria. Earlier, we saw that for each cell, we had information about how many counts were there in that cell or that barcode, and how many um, features or genes we observed in that barcode, us for that barcode. So to that, we are also adding the information about how many um, what proportion of counts are associated with mitochondrial genes, which we are identifying using this pattern in the gene names. So our genes, they have, so for example, I think if we do row names of the data, we will find the names of genes there, of which some will start with empty minus or empty dash, which are the mitochondrial genes. So we can identify them using this pattern matching. So these are the genes associated with mitochondria, and we want to identify them and filter them out. So that is what we are specifying in this function, percentage feature set. And it adds a column to our data object, which is percent underscore empty mitochondria, which was not there earlier, but now it is there. And this is the proportion of the fraction of genes associated, a fraction of counts associated with mitochondrial genes. There are other options in the percentage feature set function, such as you can, in particular, provide a list of gene names which you want to look at. So if you are there, maybe not all, in the system that you are studying, maybe not all mitochondrial genes have names starting with empty dash, or maybe there are other genes also which have names starting with empty dash or some other things. So you can, give it a list of mitochondrial gene names, and then in this, option features. And then next step, what we are doing is we are making a violin plot for these things. Let me actually open the output of this. So I have already run all this in, before the workshop, and I will just show you what the output here is. So there are, what we are doing is in the next step, we are using the violin plot function from the this package to make 3 violin plots representing the distribution of the number of genes observed in each of the barcodes associated with each of the barcodes, and total counts associated with each of the barcodes, and the percentage of mitochondrial counts. So it looks like, there are some barcodes which have all the counts, majority of the counts, close to 100%. In each dot here is a cell. So the majority of the counts in some of the barcodes is mitochondrial. So we certainly want to filter them out, because let's say we are interested in the mRNAs from our nuclear DNA. So we will set a cutoff close to, let's say, 15-20% or something like that. And then also, again, this is, not the best data for our purpose, but I apologize for the choice. Here they went for very deep sequencing of the data, close to 1 million reads, I think, per cell, or something like that. And after deduplicating, they had UMI counts very high. In general, you will have lower UMI counts, close to, for droplet-based methods, we typically currently see order of 3 to 4,000 UMI counts, sometimes maybe 6,000, 10,000 if you do very deep sequencing, but certainly not this high. So here we will set a higher threshold, close to something like 950,000 or something. And also, it looks like there are a lot more genes observed per barcode than is typical of single or droplet based methods. But we will set a cutoff, somewhere between 200. Let's say what cutoff we have set here. So we are specifying in the next step, based on these diagnostic plots, we are specifying cutoffs based on which we will filter out some of our cells. So we will remove those barcodes which have the number of genes associated with them greater than less than 200. So we will remove everything which has less than 200 genes in them, which have less than 950,000 counts, or the mitochondrial gene percent is greater than 20%. We will filter those out. So that is what is going on. And we end up with this violin plot where we have filtered out the undesired or poor quality cells. You can also look at the scatter plot of two of these characteristics or quality metrics together, such as here we have plotted percent mitochondrial content and the count of RNAs. Generally, you will expect that, I think the expectation is that, let's go to the Surat Vignet. You can load this for, so the expectation is that as the total counts of in a barcode increases, the total number of genes observed in that barcode will also increase, and the cells with very few counts may have higher mitochondrial proportion. So those are some typical observations, but they may differ depending on your data. So N feature, thank you for the question by John. It is an important question. So what is the difference between N feature underscore RNA and N count underscore RNA? So count is representing the count of RNA is representing the total UMI counts in a barcode, and N feature underscore RNA is the total number of genes. Features are for genes. So total number of genes associated with a barcode. I will repeat. Now N feature RNA is the total number of genes, and N count RNA is total UMI counts in that barcode. So typically in droplet-based data, you may need to also have a higher threshold. So we may want to say that N count underscore RNA less than something also. As for example, I think here they do. Based on their diagnostic plots here, they find that there are some barcodes which have a very high, total UMI counts. So you want to filter them out by saying that, or there may be very high total number of genes observed. So you may want to filter them out because they may be doublets, where two cells have fused together or they are in the same drop, and you want to filter them out. So you may have an upper threshold on the total number of features that you want to keep. So what I did here, someone asked for, can you show this? I will just show it here. This is not something you will need. I just use it for demonstration purpose. Row names is getting the row names of the count matrix, which are the gene names, and that I'm subsetting to get using this pattern matching function grepo to identify the names associated with the mitochondria. This is not a part of our R Markdown document. Any questions on this? So I think we are still doing good on time. We will meet our goal of getting to normalization and feature selection today.
Hey, Krishna, can I ask a question?
Yes, go ahead, please. Thank you.
For this plot of n feature versus n count on the Sora website, sort of scatter, if you sequence very deeply, would you eventually expect this plot to tail off where you sort of saturated the number of features you could detect, and even as you increased the counts?
Yeah, that will happen because there are only so many genes that we have annotated in our references. So unless and there are also only perhaps so many genes. So maybe there are 30 plus thousand genes in our human genome. So well, that will be the number that you will see at max. And if you sequence very, very deeply and you observe all of them in every cell, then you will see basically a single number up here.
Okay, thanks.
Any other questions? Thanks for the question. All questions are welcome. So one, so there are some considerations here, in particular, regarding these cutoffs. These cutoffs are based on heuristics and subjective examination of diagnostic plots. And there are some, questions that have been raised around them, and there have been many, many tools developed for doublet identification, in particular. Also for empty drops, I believe there are several tools out there. So there are, and they utilize, different aspects of the data to do quality control, and I'll just discuss them briefly in the upcoming slides. So first of all, regarding the mitochondrial percent cutoff, there are, what we did currently was based on our examination of the plot. We just set a cutoff based on something like 20%. But people have also studied, in particular in this study, Osorio and Kai, they published different or recommended cutoffs for different cell lines and cell types. In particular, the motivation for setting different cutoffs for different kinds of systems is that what is, let's say, if let's say you say observe 30% mitochondrial counts in cardiomyocytes, that may be normal because cardiomyocytes are very active, and it has been observed that they have a much higher mitochondrial proportion than, let's say, lymphocytes, in which if you observe 30%, maybe those lymphocytes are stressed. So you may want to optimize your cutoff based on your understanding of the system that you are studying. And also there is this one reference, but there may be others also, where in particular people have folks have reported that humans appear to have a higher proportion of mitochondrial counts than mice. So maybe set a higher cutoff for humans. And they have also provided reference values for a range of mouse and human cell types, as I've shown here in the plot on the right. So you may want to study that and determine some your cutoffs more judiciously. And again, you can find the references in the notes on the slides. And then there are other approaches which are which are adapting these cutoffs based on the data itself. And they have an approach such as, instead of manually setting the cutoff, what they do is they look at the quality metrics, and they choose they filter out cells which are greater than 3 median absolute deviations away from the overall the median of the cutoff, median of the quality metric. So let's say for the mitochondrial proportion, you have the Violin plot and you have the median of the distribution, and all the barcodes which will have mitochondrial proportion greater than 3 median absolute deviation away will be filtered off. So scatter is the bioconductor package, I believe. And there are different approaches out there. For doublets, how do we identify them? So the approach that we just discussed was based on UMI counts. First of all, why doublets happen? Just because there are some drops in which multiple cells may come together. Biology is messy, and so is chemistry and all the experimentation. But there is an assumption there the assumption is that um, all cells, all cell associated barcodes will have a uniform distribution of UMI counts. That is the assumption, kind of, which may not be true because some cells may be much larger than the other cells, and they may have just higher UMI counts because they are larger cells, not because they are in a drop with other cells. So there are other tools which have been developed for filtering of doublets, such as Doublet Finder from, again, I think Gartner Lab at UCSF, and Scrublet. Don't remember where that is from. But what they are doing is they are simulating a doublet profile. What they do is they randomly pick cells from your population and they create a simulated doublet expression profile. So assumption is that most of the cells will have two different kinds of cells, and then when you most of the doublets, which are real doublets, they will have two different kinds of cells coming together, and their expression profiles will be a mix-up of two cells. So if you randomly sample from your population two cells, they are more likely to be single cells because hopefully there are very few doublets in your overall population that you are studying. And they simulate these doublets, and then they compare the observed expression profiles in all the barcodes with these simulated doublets, and anything that matches the simulated doublets within certain criteria for significance of matching, etc., are removed as doublets. I hope that makes sense. But again, this is not a perfect method either. um, concerns have been raised that some mixed lineage cell states may be present in your population, which will manifest hybrid transcriptomes. So, for example, one cell line are differentiating and progressing towards other cell type. One cell type toward moving towards another cell type may have some in between cell state, and may manifest some mixed transcriptome. So, well, in that case, simulated doublet approach may not work. So again, people have developed many methods out there, some based on um, deep learning based method or other metrics, or deconvolution instead of simulating doublets by mixing two cells together. Instead, use some reference databases to perform deconvolution of cell expression profiles and check if they are matching mixed types. And well, there are, I think, dozens of tools out there right now. So just for your knowledge, if you want to look into the literature and choose some other tools, you can do that in your own time. Also, another concern which has been recently raised last year in this publication, I think this is a group from Berkeley, where they reported that some of the drops, instead of having one single cell or one single bead, which is also the goal, each drop has one bead and one cell. Some of the drops may have multiple beads because, again, we are creating drops from things or containing things, and things may not go perfectly. So, which they called as barcode multiplets or bead multiplets, I guess. So another thing that may happen is that the same bead, they found that may have multiple barcodes. The claim from 10x, ideally, there would be one single barcode on each bead, but there may be a mix-up of barcodes because, again, engineering is messy, so things may have some beads may be imperfect. So in that case, what we will end up with is the counts from one cell will be get fractionated or divided into two different columns in our counts matrix, because they will be associated with two barcodes, and one cell will lead to appearance of two cells. So that can again be misidentified or mischaracterized as some rare cell population or some new fancy biology going on. So, well, currently most analysis pipelines, especially in single cell RNA-seq, do not account for this. I don't think there are any methods that have been proposed for single cell RNA-seq, but for single cell attack-seq data, Cell Ranger, after this publication was in the preprint or something, they borrowed the method reported in this approach, and they implemented it in the Cell Ranger pipeline also. So Cell Ranger does removal of barcode multiplets for single cell attack-seq. I don't think it does for single cell RNA-seq. So you should be just careful or be cautious, or just be aware of this thing also out there. Now, a few other considerations on this discussion. What we worked with was mouse data. So we had empty dash for to identify mouse mitochondrial genes. For humans, you may the genes are named in uppercase letters, so you may need uppercase empty dash. And finally, there is another consideration which you may want to note. That is that dead or decaying cells may manifest high expression of certain genes, such as MALAT1. MALAT1 is generally highly expressed, but it has been found, and 10x Genomics reports that sometimes you see very high levels of MALAT1 in your cells, and that may be because for reasons other than some new biology going on, such as decaying cells or dying cells, etc. So try to give you some more information behind the quality control than what is in the standard workflow, so that you are aware and you can implement them your own time. We can certainly not do all that right now. Any questions regarding that? Looks like we may have to divide our normalization discussion between today and tomorrow, or maybe we will be able to cover it all today. But if you have any questions, please feel free to ask. I will I think we can manage our time tomorrow to discuss, cover everything. Okay, so the next step after we have done all this, what we have done thus far is, we demultiplex the data. We did not do it, but we discussed it. We loaded the data in Surat, and then we did the quality filtering. Now, hopefully, all of our barcodes are single cells, and we have genes that are of interest to us, or which are about which we can say something. Now, the next step is to normalize the counts. And the reason why we need to normalize is because there is a variation in sequencing depth across single cells. So for example, some cells we will have, let's say, 10,000 total reads or UMI counts, while in some others we will have 200,000, or maybe for whatever reason, we may observe that. And 10 out of 10,000 in cell 1 is not the same as 10 out of 200,000 for a gene in the same gene in cell 2. So we are looking at the expression profiles, and that is actually in some sort of a, we have to look at the UMI counts in relation to the total UMI counts in a cell. And that is also illustrated in this figure on the right. In general, we expect that the normalized expression of a gene should not depend on sequencing depth. Yet, when we, in this figure, what they did was they bend the genes into 6 categories based on their mean expression level. So for example, here you can see the y-axis ranges from 4 to 26, and here it ranges from 10 to the power minus 5 to 10 to the power minus 3. These are all non-log scale, I believe log 2. But anyway, when they bend these genes into different categories, and then they looked at how the gene-wise UMI count was varying for each cell, across with respect to the total cell UMI count. So each dot in the scatter plot, which they created, was a single cell, and for each cell we have the UMI count of the gene in this of what gene in this group, and plotted against the total UMI count for that cell. So there appears to be a trend. Cells which have a lot more UMIs have a lot more count for any of the genes. So that is not ideal, because that is not true signal. The gene expression does not depend on the sequencing depth. So we need to normalize the data. The one way to normalize, or maybe to just divide the total counts for a gene, the UMI count for a gene in a cell, with the total UMI count in that cell. But that is also not normalizing the data properly. We can see that there are different trends for different categories of genes. So, here for this group, it still looks like there is so some of the genes are not uniform. Like there is ideally we would have a uniform or a horizontal line kind of distribution like this, which means that the gene expression is not varying with the cell UMI count, but we may have patterns, raw counts. It seems like scaling of counts relative to the total UMI counts is not adequate either. So what we one of the common approaches for normalization is based on log normalization. And the way it goes is you take your you first divide the gene count in a cell by the total UMI count in that cell. Then you add a pseudo count, which is a number like 1 or 2 or something, a small number, and then you log transform. The reason to add a small number is because log of 0 is not defined. So we have to add at least some minimum value of 1 or something. So 1 is a popular choice, I believe. So you have the total UMI count is 1 plus something, and then you log transform that. So this is inspired by this approach is common also in bulk RNA-seq analysis. There are some reasons which motivate this, in particular in bulk RNA-seq, but the same motivations also prompted its use in single cell RNA-seq. So for example, one of the motivations is that in general, the gene counts may range over several orders of magnitude. Some genes are very poorly expressed, some are very highly expressed. So we may observe a range of distribution. And if you directly compare these large numbers, then it's not ideal, it's not helpful. So we want to compare them in terms of log fold change. Also between different samples, some samples may have very high expression of a particular gene because let's say some genes are expressed in lymphocytes while some genes are not expressed in monophils. So you will see a very large range of distribution. So anyway, for those purposes, log normalization is popular and useful. And also in some cases, and it is common, actually, that the variance of a gene's count is proportional to its mean expression. So log normalization again prevents highly expressed genes from dominating the analysis. So those are some of the reasons that have been given for using log normalization in bulk RNA-seq. There are more, I believe, but these are some. So for these reasons also, UMI log normalization was used in single cell RNA-seq and continues to be used in single cell RNA-seq. Single cell counts, especially at the reads level, they have a very high, they have a wide range of distribution over orders of magnitude. So log normalization was adopted, but UMI data do not have that wide range of distribution, but still log normalization is done. In practice, some authors have reported that this is useful and a good choice, a good starting choice for a normalization. But there have also been recent developments which have criticized log normalization and provided alternatives. So scRNA developers in their standard workflow, which we are implementing also today, they have log normalization implemented by default. But they have also recently published a paper where they have pointed out the pitfalls of log normalization and also reported another method. So log normalization also, what they found in this recent development, that it is also not getting rid of that, pattern of the pattern of normalized counts with respect to the cell UMI count. So it is still not normalizing in the sense that normalized expression is not independent of the sequencing depth. So that's a problem. And also what they did was to further look at that the problem, or what they did was they looked at the same data set and compared it with its they collected one data and they downsampled it. online, like using digital approaches or programmatic approaches. They downsample the data, which is basically to say that they threw out some of the counts, and they compared a downsampled data with itself, and they still saw that there are lots of they found, actually, when comparing these two data sets, they found that there are greater than 2000 differentially expressed genes that could be identified, which is basically the same data compared with its downsampled version, which should not happen. So it means that log normalization is not properly normalizing counts. And also in plots such as UMAP plots, which we will be discussing tomorrow, you can see that there is a separation of cell populations. So, well, for those reasons, they proposed another method. I will skip this slide for today. We can come back to it tomorrow, but I will describe what the normalization approach, their normalization approach is. So first, they start with, there are three steps. First, they start with looking at each gene, and each gene is receiving some contribution of, to its counts from the total UMI counts in a cell. So we have for each gene. So each plot here is for in the top, here, each of these plots is for a gene. Um, and we have the UMI counts for that gene in a cell, and also the total counts for UMI counts for that cell. So that is, those are the two axes in each plot for each gene. We have each plot, each dot here representing the UMI count for a gene, and the total UMI count for the cell. And each dot is a cell. And what they do is a generalized linear model fitting, which is which is a variant of the ordinary linear fitting that you may have been doing in Excel or wherever else. You fit a line, but the error distribution is different from the ordinary linear regression. It is here assumed to be negative binomial error. So that is the what is going on. And again, I'm happy to answer more questions. I think we can save the questions for tomorrow on normalization, and we can revisit it in the beginning tomorrow. But after you have done that, what you get is the an intercept term for each gene, slope term for each gene, and also a dispersion parameter for the negative binomial error distribution. And you do that for all of the genes in your data. And then finally, they make, and then the next step, they make, a plot for each of the, they do it all numerically internally, but to illustrate it, we have a plot where, again, on the x-axis, we have the gene-wise mean, the abundance level of each gene in the population. And on the y-axis, we have its learned intercept, slope, and dispersion parameters. And the problem with what we can see is here is that what they found is that these parameters are not very stable. These parameter estimates for each gene, based on this analysis that we did here, is not very stable. So if you look at different subsets of cells, compared to all of the cells, instead of all the cells, you look at a subset of the cells and then look at the another subset again. Ideally, what you would expect is that beta naught, beta 1 will be the same value, looking at different subsets. But they found that this is not the case. So there is a need for stabilization of these parameter estimates. And the way to do it is by imposing more assumptions on the data. And some of the assumptions are that most of the genes are not differentially expressed, and that genes with similar abundance levels have the same contribution receive the same contribution to UMI counts by the technical factor of total sequencing depth for a cell. So that's what they are doing. For here in this plot, each dot is a gene, and for each we have its learned parameters. And by fitting a trend line through this plot, they are identifying for each gene, given its mean expression level, we can now predict what the contribution of sequencing depth is to its count level. And then we can deduct it and scale it, with respect to the variance of with respect to the dispersion parameter of the fit to get what are called as Pearson residuals. So these are normalized values, and what they represent is now, after normalization, the values range from negative values to positive values, and in practice there could be anything, but the expectation is that the normalized counts will be such that for genes which are not differentially expressed across the population, the mean will be 0 and the variance will be 1. And for genes which are differentially expressed, we will observe a higher, a very high, a high variance, and that we can identify as differentially expressed genes. So that is what is illustrated here, and they verified it in various ways. And in particular, they observed that after doing this kind of normalization, comparing one data set with its downsampled version returned only 11 DE genes, which is much fewer than the 2000 from based on log normalized data. So that is what they recommend in Surat now. You can run this line of this code chunk at home, or this is just one code chunk to log normalize. We will pick up our discussion from here tomorrow, and I will review this again tomorrow in the beginning, and also everything that we have discussed thus far. So you can ask questions regarding that. Feel free to log out now. I don't want to take too much of your time, but also if you want to stick around and have any questions that you would like answered today instead of later on. We also have 1 hour reserved for just questions on Thursday, so you can also ask your questions then. But if you want to ask something now, I'm happy to be here with you and answer them. Well, once again, thank you for your attention today. We won't discuss the next slides today, and we will start with everything again, just a quick review of everything again tomorrow. And tomorrow we want to discuss what it means to reduce dimensions of the data. Things like UMAP and what they are doing, and TSNE, etc., which have become popular. I'll just scroll through the slides, and clustering and why we are implementing certain clustering algorithms for this as others, etc. And those are our topics for tomorrow. Any questions on what we have discussed thus far? I hope the discussions have been useful, and again, if you have any questions, please feel free to ask or allow off. So how to upload files from Geo? It is, well, I will just quickly answer that question. So Geo is a website where people often deposit their data. You will have to first of all download it. So you can go to, first of all, you have to the way. Let's see if there are. So you go to you will find the identity for geo identifier for your experiment of interest reported in the paper that you are interested in, and you will have these geo identifiers. You want to save these identifiers in a file and then use what is called as I think SRA tools to download data from the Geo website. See, so prefetch and FASTQ dump. Prefetch will fetch the data, and FASTQ dump will convert it into the FASTQ format. And I think we cannot demonstrate it all of it here. There are some links in our introductory RNA-seq workshop. At the end, you will find one slide discussing some things about that. In particular, I can one thing I found that, is that this FASTQ dump is kind of slow. It doesn't really, sometimes it fails when you are downloading a lot of data. So you can look into parallel FASTQ dump. For those kind of, I think in general, I've benefited from using parallel FASTQ dump. So, well, that's the one I would recommend. And the steps are pretty simple, from you just have to run things on the command line, and hopefully you attended our introductory RNA-seq workshop, because all the tools, once you have to run them from the command line on Winton, once you have the installations, I believe some of these things are perhaps already installed on Winton. But once you have the installations, it's all about, naming the files and some run parameters. And you can again look at our introductory RNA-seq workshop for more details. Thanks for the question, sir. Any other questions? Oh, well, folks, sounds like or looks like, we are done for today, and we'll see you tomorrow. Thanks for your attendance today, and it's been a pleasure discussing. So I'll log off now and see you tomorrow.